A very good morning to all. Today I am going to discuss about nitrogen metabolism. First of all, the introduction. Nitrogen is an important element for plant life. It only comes next to carbon, hydrogen and oxygen. There are various sources of nitrogen. First of all, molecular nitrogen, which is available to only few living organisms. Nitrate, which has to get converted into ammonia before entering the metabolism. Ammonium compounds, of which ammonium ions are absorbed directly by the roots of the plants along with the nitrate ions. Organic nitrogen, which is present in soil in the form of proteins or partially destroyed proteins and which can be absorbed by plants as amino acids, ammonia or nitrate directly. Nitrogen metabolism is defined as the series of biochemical changes taking place inside or outside the plant body which result in the construction of complex nitrogenous food from its simpler derivatives and the destruction of complex nitrogenous food into its components. It is one of the most important aspects of the plant life on which also directly or indirectly depends animal life. The series of reactions of nitrogen metabolism make a sort of cycle, and the supply of nitrogen from atmosphere and its return to the atmosphere is termed as nitrogen cycle. This cycle mainly consists of nitrogen fixation, ammonification, nitrification and denitrification. Here you can see the diagrammatic representation of nitrogen cycle. The nitrogen comes from the atmosphere, it goes into the soil where various decomposers are present in the soil which convert nitrogen into nitrates, nitrites, and they get absorbed. These nitrates and nitrites get absorbed by the plant roots, or there is present a symbiotic bacteria in the roots of leguminous plants which also converts atmospheric nitrogen into ammonia or nitrites which get absorbed by plants. And again, when decomposition occurs, this nitrogen is again sent back to the atmosphere. So nitrogen fixation, it can be defined as the phenomenon of conversion of free nitrogen into nitrogenous salts to make it available for absorption by plants. Nitrogen fixation is basically of two types. One is physical nitrogen fixation, another is biological nitrogen fixation. Nearly 200 million metric tons of nitrogen is fixed annually by natural processes. Only 5% of this natural fixation occurs by physical processes. Physical nitrogen, which is called as physical nitrogen fixation, and about 95% occurs through microorganisms termed as biological nitrogen fixation. Physical nitrogen fixation, it starts with combination of atmospheric nitrogen with oxygen under the influence of electric discharge and thunder to produce nitric oxide. Here you can see the chemical reaction. Nitrogen get combined with oxygen under the influence of electric discharge and thunder, and there is formation of two molecules of nitric oxide. This nitric oxide is then oxidized to nitrogen peroxide in the presence of oxygen. The chemical reaction shows the two molecules of nitric oxide combines with one molecule of oxygen and it forms two molecules of nitrogen peroxide. During rains, the nitrogen peroxide combines with rainwater to form nitrous acid and nitric acid, which come to ground along with rain. The chemical reactions also shows that the two molecules of nitrogen oxide combines with rainwater and which give rise to one molecule of nitrous oxide and one molecule of nitric acid. On ground, the alkali radicals of the soil react with nitric acid to produce nitrites and nitrates which are soluble in water and which can be absorbed by the plants through roots. These nitrates are either of calcium or potassium salt. Coming to the biological nitrogen fixation. The biological nitrogen fixation is categorized into mainly two types. One is symbiotic nitrogen fixation and another is asymbiotic nitrogen fixation. First of all, we will discuss about symbiotic nitrogen fixation. This type of nitrogen fixation is carried out by bacteria frequently found in the root nodules of leguminous plants. The most common bacterium is Rhizobium. Two types of strain of Rhizobium are present. One is fast grower and another is slow grower. There are various examples. The fast growers include Rhizobium leguminosarum, Rhizobium trifoli. The slow growers involve Rhizobium japonicum and Rhizobium lupinii. Rhizobia are commonly found in the soil of crop fields where bacteria and plant both specifically affect each other's growth. The symbiotic nitrogen fixation continued. The nodule formation occurs, which is the main process in case of symbiotic nitrogen fixation. Development of nodule is rather a complicated process. Initially, host cells have to recognize the correct strain of symbiotic bacteria. This is controlled by the specific proteins found in a host cell called lectin. These lectins interact selectively with microbial glycoproteins found in the capsule of bacteria. Here, serve as determinants of recognition or host specificity. Moreover, calcium plays a critical role in modifying the ability of root to absorb rhizobia. Nodule formation is initiated by the infection of root hair by free-living rhizobia. These bacteria cannot digest cellulose, hence they enter the root hair through the tips where cellulose is nearly absent. Leguminous plants release tryptophan in the soil, which is absorbed by the rhizobium and is metabolized to indole acetic acid. The rhizobia produce another characteristic substance called root hair curling factor that causes deformation and twisting of root hairs. The root hairs gets penetrated by a large number of rhizobia, which collectively convert into an infection thread lined by cellulose and which is provided by the host cell. The infection thread intrudes, settles and liberates its contents in a cortical cell, which is usually a polyploid and generally tetraploid. The cells of bacteria enlarge, assume pleomorphic shape and are called bacteroids. These bacteroids stimulate polyploid as well as surrounding cells to divide, which ultimately form the cortex of the nodule. The nodule contains a pink colored leg hemoglobin pigment which, like true hemoglobin, combines with oxygen and carbon dioxide and gets readily oxidized into brown form with a trivalent iron. The development of leg hemoglobin as well as nitrogen fixation capacity are outcome of symbiotic association. Individually, not there occurs the development of leg hemoglobin as well as there is no nitrogen fixation. This occurs only because of the association between the rhizobia bacteria and the leguminous plants. The process of nitrogen fixation, whether in symbiotic or asymbiotic forms, involves the reduction of atmospheric nitrogen to ammonia by the enzyme nitrogenase. Nitrogenase is extremely sensitive to oxygen. The leg hemoglobin protects the nitrogenase enzyme from oxygen and simultaneously makes oxygen available to bacteroids for ATP production required for nitrogen fixation. It is the diagrammatic representation of the formation of fruit nodules of leguminous plants. Here you can see the bacteria intrudes inside the root hairs through the root tip where it forms an infection thread by releasing a root hair curling factor which degrades the root for their entry, and ultimately the cortical cells and polyploid cells, they divide and form thick, large number of cortical cells forming a root nodule. Coming to mechanism of nitrogen fixation, the fixation process is carried out by enzyme nitrogenase. Reduction of nitrogen into ammonia depend upon availability of ATP and reduced substrate capable of donating hydrogen atoms to nitrogen. ATP is generated in bacteroid respiratory chain system. Reduced substrate, that is glucose 6-phosphate, is obtained from the host cells. Reduced NADP together with ferredoxin function as electron carriers. The ATP interacts with non-heme protein component of nitrogenase and brings about conformational change to convert it into a powerful reductant. This powerful reductant then transfer electrons to reduce nitrogen into ammonia. 4 molecules of ATP are hydrolyzed for each pair of electrons transferred to nitrogen. Thus, reduction of 1 molecule of nitrogen into 2 molecules of ammonia requires 12 molecules of ATP because 6 electrons are required per molecule of nitrogen reduced. Here you can see the chemical reaction. 1 molecule of nitrogen requires 12 ATP, 6 protons to produce 2 molecules of ammonia, and these 12 ATP get converted into 12 ADP molecules and 12 inorganic phosphate. Now coming to asymbiotic nitrogen fixation, many free-living bacteria, fungi and blue-green algae are capable of fixing free nitrogen. Bacteria are further divided into anaerobic forms, which include Bacillus polymixa, Clostridium species, aerobic forms, which include Azotobacter, Bergerichia, photosynthetic forms, including Rhodospirillum, Rhodopseudomonas, chemosynthetic forms, which involve Thiobacillus and D-sulfur vibrio. There are various types of fungi which are involved in asymbiotic nitrogen fixation, such as Bullaria and yeast. The blue-green algae which are involved in asymbiotic nitrogen fixation are basically unicellular forms, which include Gliotheca, Senicoccus, filamentous non-heterocystous forms such as Oscillatoria, Lingua, Formidium, filamentous heterocystous forms which include Nostoc, Anabina, Cytonema and Calothrix. Nitrogen in the soil. Normally, plants absorb nitrogen from soil in the inorganic forms, either as nitrate or as ammonium ion. Nitrate is first reduced to ammonium level in a series of steps in order to be absorbed by the plants. Various processes rendering soil nitrogen adsorbable to plants are described as under. First of all, the denitrification process. The process of conversion of nitrates and nitrites into ammonia, nitrous oxide and nitrogen are defined as denitrification. Ammonification, the process of release of ammonia, is known as ammonification. Nitrification, the process of further oxidation of ammonia to nitrates, defined as nitrification. Then comes the nitrogen assimilation. The atmospheric nitrogen is fixed in the soil in the form of nitrates, which are absorbed by the plants and converted into ammonia through a series of steps catalyzed by various enzymes. The enzymes involved in the process are nitrate reductase, whose main function is to reduce nitrate into nitrite. Another enzyme which is involved, it is nitrite reductase, which converts nitrite into ammonia, and the final product ammonia thus combines with organic acid to produce amino acid. Here we finish with our topic nitrogen metabolism. Thank you for your patient listening.